Right, welcome everyone on Zoom joining us live, and over on YouTube we have currently 14 people over there. Today is a special CNS seminar event because we teamed up with the journal Brain Structure and Function and bring you their special issue. So you're in for a treat of 2 days extravaganza on the structural connectivity of the cerebral cortex. And in total, we have 10 speakers, and we have the 2 editors of the special issue with us today, who will be running the show. So I'm happy to hand over to Hiromasa to introduce our session and the speakers. And you see me again in between to take your questions. And I'm also taking your questions over on YouTube. So if you want to write in the chat over there, I can bring the questions into Zoom. With that, over to you, Hiromasa.
Thank you. So hello, everybody. Thank you very much for coming. So this is a symposium kind of jointly organized by CNS seminars, clinical neuroanatomy seminar on the brain structure and function. So my name is Hiromasa Takemura. I'm an editor in the brain structure and function. So this symposium is kind of focusing on structure connectivity of the cerebral cortex. So, well, I think the original aim of this symposium is nowadays, as you may know, I think it's very difficult to organize an in-person conference. I think people cannot travel. Then I think people miss the opportunity to talk about some latest scientists. But I think still the topic on structure connectivity is quite important to understand both neuroanatomy and brain function. So we are just going to highlight the latest topic of the structure connectivity by inviting author of the recent brain structure and function special issue of the structure connectivity, which cover a wide range of the topic of structure connectivity measured by many different type of methods. So please join the discussion because the talk is pre-recorded. I think everything should be on schedule, but we always have 5 minutes question time for all talks. Then please free to use your time microphone to ask question to speaker. And we also have a question session at the end of the day. So you can ask any question to any talk at this time. Then Steph will moderate discussion session. Then you can also ask question to speaker using chat function. And in this case, you can either ask question during the talk or after the talk. So this is a schedule today. So we have 5 speakers about the structure connectivity. Then at the end of the all talks, so we have 25 minutes for discussion to every talks. So thanks, and please join and enjoy. Do you have anything to add, Michelle? Fine, yes.
So Michelle is in charge of playing the talks.
Hello,
Sure.
my name is Guilherme Fresh, and I would like to, on behalf of my team, to thank the CNS Seminars organization, along with the Brain Structure and Function Journal for organizing this virtual symposium and allowing us to present our work. Our work is entitled Principles of Temporal Association Cortex Organization as Revealed by Connectivity Gradients. We would like to take a moment to mourn the loss of Karl Zilles, Dr. Karl Zilles recently, who left a huge contribution in our field, including founding the journal, organizing this symposium today, but also to acknowledge his contribution, this particular contribution to the ideas that we're going to present here today. So in 2002, there's a paper by Passingham and others called the Anatomical Basis of Functional Organization in the Cortex. And in that paper, they describe that they suggest the term family for a cluster of areas that share a similar pattern of connections. We take the term from Zilles and colleagues, who noted that related areas can be grouped into neurochemically similar families on the basis of receptor mapping. So, of course, at this time, he was borrowing the idea of defining clusters of regions in the brain defined by receptor mapping to establish clusters of connectivity, which was actually defining these regions in the brain by their connectivity fingerprint. In this case, he was using these spider plots showing the efferents and afferents of each region, and if when traversing the brain, if a given point would have a very different connectivity pattern than another, it would be deemed to be in another region. So you would have this notion of defined clusters of connectivity throughout the brain. So this idea of gradients of connectivity in opposition to clusters of connectivity in brain has been gaining traction in the last couple of years for a couple of reasons. First, it actually enables you to discover the gradually shifting and overlapping connectivity patterns in the brain, in opposition to just saying this region is different from this region. This type of work has been done in a more global fashion with the seminal work by Margulis in 2016, describing the transmodal to unimodal gradients in the whole cerebral cortex. It can also go to more system-specific analysis. For example, the Kuhn-Hack's work in 2017 showing the two modes of the eccentricity and polar angle modes of the visual cortex showing up in an analysis with only resting state fMRI data, not necessarily using a task. Now, the cool thing about gradients is that they actually reveal to you the principles of organization, so the directions of these connectivity changes. So you're no longer describing a relationship of this region is different from this region, but now you are putting all these regions or all of these voxels even into a continuum into how different or similar they are in several different dimensions, which we call principles of connectivity. Now, we wanted to study gradients of structural connectivity because we believe that gradients of connectivity may provide a good basis for biology-based models of the human brain. And so if we are to use these gradients, we wanted to study the physical implementation of these gradients of connectivity and not just their functional consequence. It also allows you to study the connectivity gradients in postmortem samples, which opens the way for non-human data, and also because structural connectivity is composed by a discrete set of column elements, which we call white matter tracks. We were interested in the temporal lobe as a test case for reasons that will become a bit more apparent towards the end of this talk. But essentially what we did is that we took diffusion-weighted tractography data from the temporal lobe to the rest of the brain and then applied our dimensionality reduction techniques to estimate these principles of temporal association cortex organization, which we will call gradients throughout this talk. So effectively the question we were asking was, what are the directions of greater connectivity changes in the temporal association cortex? And three gradients of connectivity, or three principles, came out. One of them ranging from more inferior to superior regions, the second one ranging from outer regions of the temporal lobe towards the posterior medium temporal sulcus, and the third one being an anterior to posterior gradient. So we found these three gradients. Now, the great thing about using this in structural connectivity data is that now we can actually ask which white matter tracks are involved in these connectivity changes, because we can back project each of these gradient values on the tracts that actually generate these gradients. And what we found is that now back projecting these gradients into the brain shows you the distinct contributions of tracts in each of these gradients. Now, if you look here, we're looking at first and second gradient, so that inferior to superior, and the second one which radiates towards the posterior medium temporal sulcus. And what is interesting is that you see very clearly why do you have an inferior to superior gradient in the temporal lobe in terms of the tracks that contribute to this gradient, and also that that second gradient that radiates towards the posterior medium temporal gyrus separates very cleanly the arcuate fasciculus contributions in relation to all the other tracks in the temporal lobe. And this is also what we saw once we actually did this for every subject and then averaged all the values of all the tracks. So essentially what you're seeing here is the values of these back projections, the 95% confidence interval of the average value for each of these tracks for each of the hemispheres in both of these gradients. So the first gradient separates basically the inferior tracks from the superior tracks, and then that second gradient isolates the arcuate fasciculus very neatly in distinction to all the other constituent tracks of the temporal lobe. the last question we wanted to address in our work was the relationship of these structural gradients to the gradual shift in functional specialization that we know exists in the temporal lobe. So what we did was that we took every gradient and we divided it into deciles, which we then decoded into Nernst length. And what we found is that that first inferior to superior gradient showed a bilateral transition from more visual terms to more auditory terms. So basically making this a auditory to visual gradient in the temporal lobe. The second gradient, the one that radiated towards the posterior medium temporal sulcus, started off in the lower ends of the gradient with more audiovisual terms, and as you further progressed along the gradient, you ended up with terms more related to language on both hemispheres, more on the left hemisphere specifically, and on the right you would have also terms related to social tasks such as facial expression, identification. Now on the third gradient, the one that radiated from the, didn't radiate, that was on the posterior anterior axis. The anterior axis was dominated by terms, by task general terms such as social, person, sentence, semantic, to more task specific terms at the higher ends of this gradient, such as recognition, face, modalities, visual word. And so with this, effectively, what we did was that we related these structural gradients to we tried to relate these structural gradients to the gradual shift in functional specialization in the temporal lobe. Now, coming back to the question of why we chose the temporal lobe. The temporal lobe has expanded and been reorganized significantly throughout evolution. So in future work, we were interested in looking at how these gradients would look in other species and if we would see significant adaptations. So as preliminary work now in a new project, we can show you that we've been working on this framework for the human, chimpanzee, and macaque looked at together. So we pick out a set of common tracts, which we then describe each point of the brain as a composition of these common tracts, and hence we can actually analyze all of these species in the same framework. What is nice is then when we apply this gradient technique to this data set again, similar gradients show up. So the first gradient, which we call the first cross-species gradient, because it puts all the species in the same framework, shows up with the same inferior to superior pattern that we saw in the human, which is interesting because that the functional specialization of that gradient was shown to be just a simple mapping from visual to auditory, and it's interesting that kind of mapping was kept throughout evolution. And then, for example, the second cross-species gradient showed again this radiating towards the posterior medium temporal sulcus, but only on the human brains, which again suggests that this mode is probably more human unique. So even in looking at just two cross-species modes, we already see one clearly conserved mode and one clearly human unique one. In conclusion, we know now that structural connectivity gradual changes within the human temporal lobe. Multiple patterns of gradual connectivity change exist within this region, and they are also essential in implementing functional multiplicity. And finally, that gradients of connectivity reveal conserved and unique connectivity signatures across species. I hope you enjoyed this presentation, and I eagerly await your questions. Thank you.
Wonderful. Now, there's quite a lot of you in the audience, so I can't see all of you on my screen. But if you have a question, you can either raise your hand in the chat and I can see that, or unmute yourself and ask your question to our speaker who's amongst.
So just one question, actually. So you don't see any projection from posterior to anterior?
Sorry, can you, so seeing any projection from posterior to anterior?
That's right, because that's what we see in monkey. It's go to the limbic, right? Limbic projection from temporal to limbic.
Oh, right, I see what you mean. So at least the cross-species analysis, that part, so that's off the paper, but anyway, so on that small snippet that I showed, we're basically just tracking
let's say, now we're no longer looking at raw projections. We're no longer looking at the raw tractography matrix. We're now looking at already the defined tracts. So we are finding these tracts, they're there. We're just looking at what is the composition of, in terms of probability of each tract being at each place. So I'm pretty sure we're finding the same projections you are. It's just the mode in which these probabilities of distribution of tracts are presenting themselves in human and macaque that are different. So, but I'm pretty sure the projections, those projections are there. They're just now, we're talking about them in terms of tracts and no longer just in raw projections. So that's also why we don't show, let's say, that back projection example again in this cross-species framework.
Okay, so there's one more question actually.
Yeah, sure.
So you said that you identified the fine tracks. So can you distinguish based on this track whether it's a feed forward or feedback?
No, not at all. So because that's a limitation on diffusion data on its own, right? Because we cannot. Well, there are some now some attempts integrating some resting state fMRI into that to try to estimate directionality of signaling. But yeah, with just diffusion, yeah, it's I'm gonna go on a limb and say that it's quite impossible. Yeah.
Thank you.
Are there any other questions in the audience?
Well, do you see any difference between left and right hemisphere? Because typically, I think in arcuate fasciculus, people report lateralization in humans.
Yeah, so we do see some lateralization effects. So namely, so there's a part where we look at the reproducibility of these maps because we're looking at 44 subjects of the human Connectome project that had 2 sessions of scanning. So we were actually, there's a part where we actually analyze if the maps hold on the second session. And what we see, and we do this for all the gradients actually. And what we see is that there is definitely there is an asymmetry effect in the second gradient, which is the arcuate caused one, in which the left hemisphere gradient actually is a lot more consistent across sessions than the right hemisphere, meaning that arcuate really drives that gradient a lot more on the left than on the right. So the gradient, let's say, shape of that second gradient is a lot more consistent on the left hemisphere, both across subjects and even within subject across the 2 sessions on the left hemisphere. Again, highlighting the role of the arcuate fasciculus in there.
Thank you.
Anyone else?
I'm going to raise a question on, good, you can hear me, okay. Let's go temporarily to the gray matter. And I just wanted to mention as an aside, I guess, there is a huge literature on gradients along the temporal lobe. I'm thinking it's as long ago as I think 2002 in before Brain Structure and Function was Brain Structure and Function. There was a special issue from Javier De Felipe. And Ichiro Fujita, in particular, at the end of his article, talked about gradients, one of which, and I see Salim is here, I'll be very familiar, was Kalbinden. It was reported as an increasing gradient from visual cortex into the temporal lobe, but you could also think of it as a decreasing gradient from the hippocampus, actually, back through the temporal lobe. And of course, there are other gradients as well. So eventually, I would guess that those 2 lines might come together.
Yeah, so yeah, I think at least one of those, the one from visual areas, I think it's clearly visible here. The hippocampal one, not so much, because so here we wanted to abstract from the hippocampus just because of the layer difference with the rest of the association cortex. So we didn't want to mix those 2. Also, there is other work on like graded parcellations of the temporal lobe, in which I think even the main author is also here. I think Claude is here, in which he included the hippocampus, and he can probably tell you that the hippocampus was definitely one of the drivers of one of those gradients as well.
Yeah, hello. Yeah, I've done similar work, but we included the hippocampus, and in fact, if I'm not mistaken, that is the prime. If memory serves, that would be sort of the main differentiation. So it goes from hippocampus to medial to lateral, and that would be the primary gradient. But that's sort of expected because the hippocampus is going to be so different, both in terms of cellular architecture and in terms of connectivity, right? So no terrible surprises there.
Lovely. Thank you very much for all those questions and comments. We're going to move on to the next talk. So I hand over to Hiromasa again to introduce the next speaker.
So we are now moving to talk 2. So next talk is by Dr. Niels Lotter. Then the title is CBPTOOLS, a Python package for regional connectivity-based parcellation.
Hello everyone, I am Niels Reuter. Here I will talk about CBPTOOLS, an open source Python pipeline for performing regional connectivity-based parcellation. Quoting Karl Zilles, a most important anatomical aspect of functional specialization is the connectivity between brain regions. Fiber tracts and their synaptic contacts constitute the anatomical basis for functional connectivity, specialization, and segregation. Segregation on the basis of connectivity, in particular the profile of connections between regional voxels and all other voxels in the brain, underlies the basis of the regional connectivity-based parcellation method. Taking advantage of these distinct connections of each area, connectivity-based parcellation, abbreviated as CBP, divides a region of interest, referred to here as a seed region, into distinct and biologically meaningful parcels. These derived parcels can be viewed as functional areas, although they are outlined by differences in connectivity rather than function. The resulting parcellations can be used to study brain functional segregation, provide a compression model for brain data, and more. Here I will outline the workflow of CBPTOOLS and provide resources for its use. It is an open source processing pipeline that uses functional or structural neuroimaging data to divide a seed region into distinct parcels, developed to make the regional CBP procedure easier to perform on large data samples. Before the workflow processing can start, first a regional CBP project must be set up. This consists of the selection of a region of interest, the resting state or diffusion data, and a CBPTOOLS configuration file. Since CBPTOOLS does not pre-process the data, it is important that the data has been appropriately pre-processed prior to using CBPTOOLS. Through the configuration, a variety of options can be defined to customize the processing, as many steps included in the workflow are optional or user-definable. Both the configuration as well as the input data is validated. Note that this validation covers only common mistakes, such as wrong parameter values in the configuration, missing data, masks and data not sharing the same space, and so on. In our paper on CBPTOOLS, we exemplify this procedure using 300 healthy unrelated subjects from the Human Connectome Project Young Adult dataset, and 3 different regions of interest depicted on the right. Shown here is a workflow diagram outlining all the steps performed by CBPTOOLS from input data to parcellation results. 3 types of input data are accepted: subject-wise diffusion MRI in blue, resting state functional MRI in green, and connectivity matrices in yellow. By being able to input connectivity matrices directly, it is possible to use any sort of pre-computed connectivity with CBPTOOLS. Included in the input are also the ROI, a configuration file telling CBPTOOLS what to do. Note that specifying a target mask is optional, as by default a whole brain gray matter mask will be used. CBPTOOLS can deal with multi-session data, as well as data in native space. For multi-session data, the connectivity matrices are averaged, whereas for native data, only individual clustering results are generated. Masks are marginally pre-processed with optional median filtering and upsampling for the seed mask, and seed extraction, downsampling, and subsampling for the target mask. The seed mask contains all of the voxels within a region of interest that are to be parcelled. It can optionally be median filtered, and when diffusion data is used, it can be upsampled. Median filtering is useful for hand-drawn masks to get rid of biologically unlikely sharp edges in the image, but is generally not recommended when masks are derived from an atlas. The target mask, on the other hand, defines the target voxels for the seed voxel to target voxel correlations, providing a connectivity profile for each seed voxel with its connections throughout the brain. Its options consist of seed extraction, downsampling when diffusion data is used, and subsampling when resting state data is used. Seed extraction subtracts the seed masks from the target mask, effectively removing within-seed connectivity from the connectivity matrix. Subsampling removes every second voxel of the target mask under the assumption that they are similar, for instance due to smoothing effects. The result is an increase in computational efficiency for computing the connectivity matrices. Next, in step C, connectivity is computed. In this step, the resting state data can be optionally pre-processed with smoothing, confound regression, and bandpass filtering. To obtain resting state connectivity markers for a given seed region, the subject-wise time series of the seed voxels and a set of target voxels are correlated. Connectivity markers for diffusion data are obtained using FSL's PropTrackX2. The ensuing data is densified and the seed voxel order is converted from F-contiguous to C-contiguous order, so that all data generated and stored during the processing is directly comparable. The PCA transform can optionally be used to reduce the number of targets, speeding up the clustering. If connectivity matrices are given directly as input, CBPTOOLS will skip processing up to step D, where one of the three included clustering algorithms can be used: k-means, spectral, or hierarchical clustering. This results in cluster labels for each individual subject. In step F, these cluster labels are grouped. First, a reference clustering is obtained by hierarchically clustering all subject-wise labels. Next, the subject-wise labels are relabeled to ensure that each subject uses the same label IDs as the reference for the most similar clusters. This allows for the subsequent creation of one set of group cluster labels by computing the mode of all labels. Optionally, it's also possible to use the reference cluster as a group result. In step E, the cluster labels are validated with metrics such as the Kalinski-Harabas score, the silhouette score, or the Davies-Bouldin index. Step G computes similarity between the individual subject labels and the group labels as a measure of validity for the group results. Options for computing the similarity scores are metrics such as the adjusted rand index or adjusted mutual information score. Lastly, various reports are generated as tables, figures, and nifty images provided as output by the pipeline. Some of this output can be seen here. In our CBPTOOLS paper, we have parcellated a combined region of interest consisting of the supplementary and supplementary motor area, considered to be a gold standard testing region for parcellation methods. On top is the seed region as derived from the Jülich Cytoarchitectonic Atlas. Note that when using CBPTOOLS, we have removed the subdivision based on cytoarchitecture. Below are the CBPTOOLS results for diffusion and resting state data. As configuration options, we configured solutions for the 2, 3, 4, and 5 cluster solutions. CBPTOOLS provides these plots from various viewpoints, although the clusterings are also mapped onto nifty images, so an image viewer can be used to inspect the parcellations as well. CBPTOOLS also outputs validity metrics as shown here. The data for these are also provided in tables, allowing you to create your own figures. Furthermore available is the interim data, consisting of the processed seed and target masks as nifty images and the connectivity matrices and cluster labels as NumPy arrays. NumPy can then be used for further processing with other scripts. Further output not depicted here are the cophonetic correlation, similarity scores between the clustering results and the user-provided reference images, the accuracy of the relabeling, and optional benchmarks that outline the execution time and resource usage of each task in the pipeline. CBPTOOLS can easily be installed from the command line using pip. Note, however, that if diffusion MRI data is to be processed, then FSL's PropTrackX2 must be installed manually. After installation, the CBPTOOLS command can be used to obtain an example configuration file and to set up a regional CBP project. Lastly, SnakeMake is used to execute the pipeline. Due to using SnakeMake, the pipeline enjoys various optimizations in its execution, as well as compatibility with certain schedulers such as Slurm. CBPTOOLS uses various immensely helpful open source packages to do most of the heavy lifting. Of note are NumPy for working with arrays, Nibabel for working with nifty images, Pandas for working with tabular data, scikit-learn and scipy for clustering algorithms and validity scores, matplotlib and seaborn for all of the plotting, and lastly snakemake for workflow management. The CBPTools paper is available as open access online, and the code is located on GitHub. Extensive documentation is available on Read the Docs, and we have also provided a set of example data to run CBPTools with. Note that the example data is directly downloadable using Cybo, but also by using datalat on the GitHub repository. How to download it is described in the online documentation. Thank you very much.
Lovely. Nias, I already saw you in the audience. If you want to make yourself visible to our...
Here I am.
Lovely. Are there any questions? No one? Well, let me start with a question then. What's the inter-individual variability between those parcels that the software generates?
That generally depends on what data you're using, also what region you're looking at. But the individual variability is reported. So there's various metrics that can be used to ultimately make a decision on what cluster solution you're going to use. So as you could have seen from one of the slides, you can select multiple values of K for the K-means clustering. Mm-hmm. So you get a 2-cluster solution, a 3-cluster solution, up to whatever cluster solution you want. And then in accordance with the cluster validity metrics and all the other reports that are provided by the tool, you can then make your decision on which cluster fits your data best. Right. I hope that answers your question.
Yeah. Okay, any other questions?
I would follow up on that with the individual or variability. Is that functional? Which regions tend to be more variable, or is it mechanical, like where they are toward the ventral surface or a midline, proximity to blood vessel?
It's a good question. I honestly don't have a clue because what I've mostly been focusing on is creating the software and making sure that that others can use it as well. So I haven't really parcellated a whole bunch of regions. I have looked into 3 regions specifically, but mostly from a functional point of view to ensure that the pipeline is reporting something that is replicating stuff that was also already published. So I wouldn't have a clue there. Although one thing I can say is that when it comes to the parcellations, it also depends a lot on the input modality that you're using. So there's the resting state modality that can be used right now for obtaining more functionally derived parcels. You can also use diffusion MRI on the other hand to create parcels based on your diffusion weighted imaging data. And these parcels don't always tend to match. There's differences between the modalities. In some cases, there's overlap, which is good, as you could have seen with the SMA. But when there is not, there's obviously something more going on that requires further investigation that can be done with tools other than CBPTools. So just to reiterate, the sole purpose of CBPTools is to help you in applying the regional connectivity-based parcellation procedure, but not to provide all the information you need to make a decision on how a region of interest can be split into several parts.
Thank you.
Anyone else who has a question for Nias right now? Can't see anyone raising a hand. If you come up with a question later on, feel free to ask the question in the big discussion round later on. So with that, I hand over to Hiramasa for our next talk.
So we are moving to talk 3 by Dr. Suji Huang. The title of her talk is High Gradient Diffusion MRI Reveals Distinct Estimates of Axon Diameter Index within different phymatal tracts in the in vivo human brain.
It's a great pleasure to present our work today on using high gradient diffusion MRI to map axon diameter index within white matter tracts in the living human brain. Diffusion MRI provides an indirect probe of structures on the micron level in tissues by using the Brownian motion of water to infer the sizes and shapes of, say, cells and axons within the brain, whose dimensions on the order of microns are orders of magnitude smaller than the imaged voxel size, which is on the order of millimeters. As such, diffusion MRI holds the most promise among non-invasive imaging methods for probing cellular structures of any depth and locations in the living human brain. Diffusion MRI benefits from having strong diffusion encoding gradients. On a conventional system equipped with maximum gradient strength, typically on the order of 40 to 60 millitesla per meter, one typically would need to play out these diffusion encoding gradients for a long time in order to accrue sufficient area under these gradients to get enough diffusion weighting. During this time, the signal is decaying due to T2 relaxation. As a result, at the image acquisition time during this echo time, the signal would be less than optimal. On the Connectome system, which we at MGH are privileged to have the original system, this maximum gradient strength of up to 300 millitesla per meter allows us to achieve this diffusion encoding gradients within a much more efficient way, such that we can pull in these echo times and boost the signal to noise ratio. In addition, having higher gradient strengths improves the diffusion resolution, allowing us to essentially probe smaller structures. Finally, the shorter diffusion times enabled by these strong gradients sharpens the diffusion probability distribution function and allows us to map out the orientations of white matter fibers much more precisely. Here at MGH, we were fortunate to have the original Connectome system, which was designed by Siemens as the ultimate diffusion machine. This is a whole body gradient system equipped with a maximum gradient strength of 300 millitesla per meter and a slew rate of 200 tesla per meter per second. We were able to enhance the efficiency of the diffusion the image encoding through novel sequence developments such as simultaneous multi-slice, which enables a 3 times increase in encoding speed. This, coupled with a 64-channel custom-built phased array receive coil, allowed us to achieve nearly 10 times increase in sensitivity for high B-value diffusion MR imaging. An important byproduct of the Connectome project and scanner was the recognition that we could extract a multitude of tissue microstructural parameters with better sensitivity using such high gradient strengths. These entities include the relative volume fractions of, say, restricted intracellular water within axons and within the extra-axonal space, compartment size estimations such as average axon diameter, and orientation distribution of axonal fiber bundles. Early work done by our group showed that higher gradient strengths improve axon diameter and density estimates. Here we show maps of axon diameter and density across the corpus callosum in a healthy subject using different maximum gradient strengths. Increasing the maximum gradient strength to the Gmax of the system improved estimates of axon diameter and density, as well as the spatial coherence of the axon diameter maps. And these results showed that higher gradient strength does indeed improve the diffusion resolution of small structures and emphasizes the importance of high maximum gradient strength for in vivo axon diameter estimation in humans. We sought to expand and extend this work to the whole brain beyond the corpus callosum by devising an optimized acquisition protocol utilizing the high maximum gradient strength, 300 millitesla per meter, on the Connectome scanner. We achieved whole brain coverage at relatively high isotropic resolution of 2 millimeters voxel sizes, and we also performed high angular resolution diffusion imaging with sampling of up to 64 gradient directions per B value. We acquired data with 2 diffusion times of 19 and 49 milliseconds and 8 equally spaced Q space shells per diffusion time for a total of 16 B values ranging from 50 up to almost 18,000. In addition, we were able to achieve relatively short echo times using these high gradient strengths and accelerated the acquisition in the slice direction using simultaneous multi-slice with a factor of 2 and in-plane acceleration with parallel imaging using a factor of 2. In total, the acquisition time for this whole brain acquisition took just under 1 hour, and we performed this in 20 healthy individuals. It's important to emphasize that it would be impractical to acquire such rich information without the latest acquisition software and hardware with such efficiency. In order to extract these whole brain axon diameter estimates, we performed the following analysis. First, we took the diffusion-weighted images, reconstructed the orientation distribution functions, and then extracted the signal perpendicular to the principal fiber direction and fitted those signals to an axon diameter model. This model consisted of 3 compartments, each of which were to represent a different component of the tissue. So we modeled intra-axonal restricted diffusion within cylindrical axons, extra-axonal hindered diffusion as Gaussian diffusion, and we also included a free water diffusion compartment to model CSF. Here, we demonstrate the ability to map axon diameters across fiber tracts throughout the whole brain in 20 healthy individuals using the Connectome gradient strengths and our axon diameter analysis. So there were a few striking trends that emerged as we performed the group average of these axon diameter maps. First, the corticospinal tracts, as indicated here in these green arrowheads, showed larger diameter axons compared to the surrounding tracts. Furthermore, there was a striking anterior to posterior gradient in axon diameter in which the frontal fibers showed smaller relative axon diameters compared to the more posterior fiber tracts in the parietal and occipital lobes. It is known from histology that the thickest axons do originate from primary motor, somatosensory, and visual areas, as established by prior studies, including this study by Innocenti and others in non-human primates. And in particular, it is known that the corticospinal tracts arising from primary motor cortex have larger diameter axons compared to the surrounding white matter, as do the optic radiations projecting to primary visual cortex. We were able to distinguish the larger diameter fibers that are known to travel within these tracts, and the trends became much more obvious as the data was averaged over multiple subjects. These findings were reproducible within a single subject, as evidenced through these separate scans that we performed on the same individual at different voxel resolutions. The corticospinal tracts seen on this sagittal view and indicated by the green arrowhead are slightly more apparent on the lower resolution and slightly higher signal-to-noise ratio acquisition acquired at 2.4 millimeter isotropic voxel size compared to the 2 millimeter isotropic voxel size acquisition. However, in both cases, we were able to distinguish the corticospinal tracts as well as the trends that we had seen in the group average. And it's important to note that although the individual subject maps were noisy, when we averaged over 20 subjects, we could see these trends become much more obvious and gain signal-to-noise ratio. So these results have led us to conclude that we can be sensitive to axon diameter in the human brain using high gradient diffusion MRI acquisitions. We know that we are just at the tip of the iceberg, however, that we're only sensitizing our measurement to the largest diameters within the axon diameter distribution, and this has motivated us to pursue the next generation Connectome scanner, which we term Connectome 2.0. So in partnership with Siemens, we will be developing a gradient system that will nearly double the current maximum gradient strength on our Connectome scanner to a Gmax of 500 millitesla per meter and triple the slew rate. And together with hardware innovations, including high sensitivity coils and multi-scale validation with orthogonal modalities, we hope to provide a next generation human MRI scanner that will allow us to map the brain across multiple scales. And it is on this note that I'd like to provide some words in memory of Karl Zilles, who was instrumental in helping us launch our original Connectome effort, as well as this Connectome 2 project. Karl was a key member of our external advisory committee, and he was a true inspiration for our efforts for multi-scale imaging and validation of human brain structure. I'd like to read a quote by Bruce Fischel, and a few words from Bruce in memory of Karl, who was a close colleague of his. So from Bruce's, in his own words, he said, "To my mind, Karl was largely responsible for the resurgence of interest in human brain anatomy. He was not deterred by the difficult and long-term nature of the study of human neuroanatomy. Instead, he put the time, energy, and resources into understanding human brain structure and built a research enterprise that made enormous progress in this laborious endeavor. These studies required patience, dedication, and appreciation for detail. His research led to a deeper understanding of the commonalities and diversity of the micro, meso, and macroscale architecture of the human brain." And it is with these words that I, on behalf of the entire Connectome 2 team, would really like to thank Karl for all his efforts and keep his memory in our hearts as we continue on this large-scale project. So in closing, I'd like to acknowledge my close collaborators at MGH, as well as generous grant funding from the NIH and the BRAIN Initiative that supported much of this work. Many thanks for your attention.
Hi, Susie.
I don't think I've spotted Susie in the audience just yet. She might join us a little bit later.
Okay. Hi, Susie.
I don't think she's here. She might join us later. I haven't seen her amongst us yet, and she hasn't responded yet. So let's hope she's joining us later on, and move on to our next speaker just now. Sorry about that.
All right. So I think we are going to talk number 4 now. Then next talk is by Dr. Kenichi Oishi. The title of his talk is Mapping Tract in the Human Subthalamic Area by 11.7 tensor ex vivo diffusion tensor image.
Hi, I am Kenichi Oishi. On behalf of my co-authors, Susumu Mori, Yuan Troncoso, and Fred Lentz, I'm going to talk about fiber pathways that pass through a small area just beneath the thalamus. But before I start my talk, I would like to thank the editors of the special issue, Dr. Takemura, Dr. Thibault Deschatin, and all related to the clinical neuroanatomy seminars. I would also like to share my memories of Dr. Karl Zilles, the founding co-editor-in-chief of the Brain Structure and Function. When I was a shy PhD student, I attended the OHBM. Karl was the chair of the scientific program committee. At the beginning of the meeting, he declared that poor English is the official language of this meeting. He said all of us can enjoy discussions. His words changed my life, and I'm honored to co-author 2 papers with Karl. So, the subthalamic area is a tiny area that locates just beneath the thalamus. This figure demonstrates the surface of the brain and the deep gray matter structures. The thalamus is shown as transparent blue. The striatum, which consists of the coated and the putamen, is transparent purple. The globus pallidus is demonstrated as transparent green. The red nucleus is shown as transparent red, and the subthalamic nucleus is shown as transparent yellow. The subthalamic area, surrounded by the yellow lines, is roughly defined as an area beneath the thalamus and medial to the globus pallidus. Although the volume of the subthalamic area is small, it contains a variety of fiber pathways. The majority of the efferent fibers project to the cortex and form the cortico-basal ganglia-thalamo cortical feedback loops. The loop also contains afferent fibers that are coming back to the thalamus, but it's not visualized here. In addition to the afferent fibers involved in the feedback loops, there are fibers from the spinal cord or the brainstem, such as the medial lemniscus, spinothalamic tract, and the tegmental lemniscus, which are combined and demonstrated as pink tractogram. And the cerebellothalamic tract has demonstrated as green tractogram here. So this is the magnified view. Note that the tractograms demonstrate only part of the fibers passing through the area. Subthalamic area is the spot where a lot of efferent and afferent fibers are converging. Understanding of the subthalamic area is important. Accumulating evidence has demonstrated the effectiveness of the subthalamic white matter intervention in treating various movement disorders. However, numerous fiber tracts pass through the small volume of the subthalamic area. Therefore, fiber pathways affected by electric stimulation or ablation of the subthalamic area are uncertain, while it is clear that multiple pathways are included in lesions or electrical fields related to deep brain stimulation. Notably, little is known about a three-dimensional trajectory of the white matter fibers that pass through the subthalamic area. The fiber pathways have been typically illustrated as a line drawing like this. Although such a line drawing is useful to understand the anatomical connections, it doesn't represent precise localization of the pathways. Histological approaches have traditionally studied fiber pathways that pass through the subthalamic area. Recent advancement in diffusion MRI enables us to delineate fiber structures in vivo. In vivo approach is particularly useful since it allows us to evaluate fiber pathways for each individual non-invasively. For example, the left figure is the coronal section of the ex vivo specimen with myelin stain. The diffusion tensor imaging can visualize the same area surrounded by yellow contour, as shown in the right figure. Let me magnify this area. Although some color contrasts might represent the existence of the fiber pathways, the image is blurred, given the resolution of diffusion MRIs with approximately 2 by 2 by 2 millimeter voxel size. Given the small diameters of the fiber pathways, which can be estimated from histological sections, accurate localization of the fiber pathways has been challenging for in vivo MRI. Here is an example. If the 2 by 2 by 2 millimeter grid, which represents the resolution of the diffusion MRI, is overlaid on the histological section, we can quickly aware that each pixel includes multiple fiber pathways. For example, the pixel highlighted by yellow contour includes part of the mammillothalamic tract, mammillategmental tract, thalamic fasciculus, and the ansa lenticularis. So, the goal of this study was to create an ex vivo super high resolution MRI atlas of the subthalamic area and to investigate the three-dimensional trajectories of the thin fiber tracts that pass through the area, hoping that the ex vivo image here enables us to bridge the knowledge gap between histological observation and in vivo MRI. The voxel size of the ex vivo MRI is 250 by 250 by 250 microns. Here is a mean diffusivity map of the specimen that includes the thalamus, lateral geniculate body, a part of the thalamus, hippocampus, red nucleus, highlighted by red, zona incerta, highlighted by green, subthalamic nucleus, highlighted by yellow, substantia nigra, highlighted by cyan, and the mammillary body, highlighted by pale green. Parts of the putamen and the globus pallidus, and here, and globus pallidus here. Putamen and globus pallidus here are also included. Here are the fiber pathways we have identified. Red bundle is the mammillothalamic tract. Yellow bundle is the ansa lenticularis. Cyan bundle is the fasciculus retroflexus. Pink bundle is the subthalamic fasciculus. The white bundle contains the medial lemniscus, trigeminal lemniscus, and spinothalamic tract, which is named the MLTL-STD complex in this study. The orange bundle is the lenticular fasciculus, and the green bundle is the cerebellothalamic tract. And here is the three-dimensional appearance of these fibers. A three-dimensional tractogram allows us to demonstrate the location of the fiber pathways in two-dimensional slices. Here are the images that demonstrate where these fiber pathways locate in the two-dimensional coronal planes. The leftmost column is the color-coded FA map, mean diffusivity map, and the rightmost column is the two-weighted image, and we can localize these fibers on these ex vivo images. The ex vivo image was transformed to an in vivo MRI atlas in the standard MNI space to extrapolate the knowledge obtained from the ex vivo tractography to the in vivo MRI. The fiber atlas is available through our website. So in this MNI space, we can localize where these fibers pass through, and we can use this color code to as a clue to identify these fibers. So in conclusion, the ex vivo mesoscopic PTI allowed visualization of the fine detail of the fiber connections, and it enables us to bridge the knowledge gap between histological observations and in vivo MRI. I would like to acknowledge my colleagues in the Department of Pathology, Radiology, and Neurosurgery. Thank you.
I can't hear you, Steph. I think Steph is muted. Yeah,
Sorry.
good.
Yes, sorry. Are there any questions in the audience?
Oh, yeah, I have two questions for him.
Wonderful, go for it.
Yeah, the first one is, why you don't talk about the field of Forel? It's a F1, F2, and F3, F1, and F2. And the second one is, can you distinguish indirect or direct projection from GPI and GPE?
So, hi, this is Kenichi. Your first question is about field of Forel. Actually, I mentioned it in my paper. It's kind of a common nomenclature of this field, but I would like to extend the field, extend the field of interest to include additional areas. I mean, the posterior portion of the field of Forel is also included in my study. So that's the reason why I use subthalamic area instead of field of Forel.
Yeah, I know that. But for example, if you look at the zona incerta, the fiber between the zona incerta and the subthalamic nucleus, it's also called a F2. So you have to mention that because that's very important. It was recognized in what's called a deep brain stimulation is based on the F2, right?
Well, yes, that's correct. I mean, I intentionally didn't use the term because I thought about the general audience. And if I start to use these anatomical terms, I thought I need to explain more about the background.
Oh, I see.
So that's the reason why I use more like a general term.
Okay. So the second question I asked is, this is based on the basal ganglia circuitry. So can you distinguish the direct projection from GPI to thalamus and GPE through the subthalamic nucleus to GPI? Can you distinguish these two pathways?
I don't think so, because what I visualized in my study is kind of thick, relatively thick fibers, and creates a bundle formation. But I am aware that many of the fibers are not forming a thick bundle, and these are crossing. So to visualize these fibers and segregate these fibers, I think higher resolution is needed. However, this is kind of a limitation of this study because currently in this protocol, the scan time was 40 hours. So to increase the resolution, I think more time is needed, like more than 1 week to get more, increase the signal to noise ratio.
Okay, thank you.
So answer your question, yeah.
Thank you for your
Okay.
question.
We had a question in the chat. Nice talk, first up. Are those diffusion MRI available for download?
Currently, you mean the 3D volume image, right?
I'm assuming.
Currently, no. One of the reasons is the copyright issue. I mean,
sorry to interrupt you. He just clarified the diffusion MRI data.
Diffusion MRI data. Yes, we are planning to do that, but at this point, not available. We are not open to this data. One of the reasons is that we are writing a book chapter using this data set. And after we clear all the copyright issue, we can open this raw data. And that's what we want to do.
Wonderful, thank you. Are there any other questions?
It's just quick question. So
sure.
What kind of software do you use for this program, for this project?
MRI Studio.
Oh, I see. Okay, thank you.
Okay, we have time for one more question. So I actually have a question for you. So these are ex vivo data, and obviously the resolution is quite high with ex vivo data. And you made the link to neurosurgery. Do you have an idea what the function of all these pathways is that you described?
Which pathway?
All of them, basically. Do we have the functions for the pathways already? Or can we just visualize them now with the data?
I mean, many of the functional studies are based on animals, mostly from macaque, and the functional MRI study is performed on human brain is extremely difficult because of the resolution issue. Because typically, fMRI resolution is kind of 2 by 2 by 2 millimeter voxel size, and that does not allow us to do fine functional study on human brain. But we do have studies based on animal models, and these, perhaps I summarized these findings on my discussion section, perhaps. So there are so many fibers. One of the well-known function is the mammillothalamic tract, because it connects the mammillary body to the anterior nucleus of the thalamus, and it's part of the puppets circuit. So it's important for the memory function. And fasciculus retroflexus is often related to mood disorders because it's connected to habenula and medial part of the midbrain.
Wonderful. I'm happy to discuss this in more detail later on. I saw that Anton also has a question. Do you just want to? There we go. Oh, hang on, you're still muted. Let me unmute you. We can't hear you, Anton.
Yeah. Thank you for the nice talk. Maybe a reference to your last question. There are a few studies, including my own, on stimulation-induced connectivity patterns in humans. So So there is there are some studies also, but of course not with that resolution as nicely shown in your data. I have a quick question. Do you have only one subject, one human data, or do you have a group of data?
At this point, I only have one data, single subject, but we do have several specimens.
What specimen do you have?
A brain specimen from multiple subjects. So we just need time and scanner to do the experiment.
Yeah. Are you planning to do that, or not yet known?
We are planning. And my future goal is to do the same thing on diseased brain to identify the change of these fibers related to disease.
Okay. With that, I think we're ready to move on to our last talk already, if I'm not mistaken. That happened quickly. So over to you, Hiroma.
Hi. So the next talk is the last talk today. So yes, the next speaker is Dr. Kathleen Loughran. Then she will talk about, well, what we can learn from the complex architecture of single axon.
So the talk I'm giving now is, of course, going to parallel very much the review that I wrote for the special topic, and that is what we can learn from the complex architecture of single axons. As I started in the text, I'm going to start with a few slides of background and then give examples of axons. This already, of course, are two examples of axons. I did not illustrate these in the text, but to some extent, this was a partial inspiration for the work that I did those 10 years plus, starting in 1988, 1989. And what we're looking at are single axons from the visual thalamus, the LGN, terminating in the cortex. There were several criteria of differences here. The parent cells were different in the LGN. Postsynaptic layers, you can see, are slightly different in the cortex. Bouton size is different. Bouton number is different. Response properties are different. And what we're looking at is the parvocellular visual cortex versus the magnocellular. And being able to identify these differences at the single axon was really very, very influential in what subsequently became known as hierarchical organization to some extent, and the segregation of visual streams, biased toward acuity or sensitivity vision. So in terms of connections, I'm going to be concentrating on extrinsic cortical, but the same questions same advantages recur in different systems, like in the hippocampus, the cerebellum. And what do single axons tell us? Well, at a very general level, are things uniform or diverse? There was a lot of interest in uniform cortical organization, but less and less evidence for that, including as the field goes to whole brain calcium imaging. With single axons, you can get exquisite quantitative parameters. You can see collateral organization, spatial organization, and to some extent microcircuitry. So how do we look at single axons? Well, in the small rodent brain, there are multiple possibilities. One is intracellular fills, another is low titer virus, a third is native fluorescence, yellow fluorescent protein or green fluorescent protein mice. In the large brain, these are still very problematic, and most of the work is is carried out by tracer injection, so a neurosurgical procedure in vivo. This is a postmortem macaque brain, but the animal had been had undergone a sterile neurosurgical procedure. In vivo, we injected tracers in visual area, allowed time for transport, then perfused the brain through the heart, collected the tissue, and processed serially. For axons, the techniques are anterograde tracers, an injection site, anterograde tracer, and then visualization of the terminal arbors thanks to axonal transport. So this is what it looks like. Here's an image of a typical injection site in the cortical area. It's about 1.5 millimeters in diameter. It's large. And you're going to get lots and lots of anterogradely labeled segments. The projection focus still looks very, very dense. And it's quite satisfying to say, okay, this is a projection in the middle layer, layer 4. But if you go to single axons, as we'll see in the next few slides, the arbors are not identical, but rather show a diversity in terms of size and complexity. This is progressively higher magnification of an isolated arbor, and you can see here the putative synaptic boutons. At the going back to the rodent, this is a sagittal section of the rodent brain with the equivalent of an intercellular fill. Here are two neurons in the motor cortex, and with serial sections, strict serial sections, you can follow the filled axons to the local. These are the intrinsic collaterals, extrinsic cortical collaterals, and then you can see multiple targets, multiple structures that are targeted by these neurons. This is a striking example of divergence, which is extremely typical of the connections. This is an exaggerated case, but all neurons will diverse one neuron to multiple postsynaptic neurons, and often to the postsynaptic targets. Single axons, this is also in rodent with low titer Simbus virus. We often talk about thalamocortical connections in one layer or another, but here the investigators were able to reconstruct individual axons. Here is the thalamic neuron. Here are the axons projecting to multiple thalamic targets. That already is a result. And when they terminate in multiple cortical areas, it's frequently in different layer combinations. Now, what I had concentrated on in the review was cortical networks in what has come to be called feed-forward and feedback. So there are various schematas of the networks. This is all three of these. Two of these are in visual cortex in the monkey, and then the filament and Van Essen summary diagram and extension of that in 1991. When we started work on this, and this was not single axon, we were able to see differences in laminar organization, such that suggesting there were at least two different systems of cortical connections. And what I'm going to suggest to you as we go on is not only are there two types, two classes, but within that, we probably need to think of further subdivisions. This is again rodent, and in the rodent there has been an explosion of techniques now for a whole brain visualization. This is from one group in 2019, but there have been others working on this. With serial two-photon tomography in the mouse, you can easily reveal diverse patterns of projection patterns of single neurons. So here is cortical thalamic, cortical medulla, and other examples. When we started this equivalent approach in the monkey, that was not possible. It still is not in the monkey. So what we did was collect strict serial sections, which is necessary for reconstruction, and then we try to retrieve and convey the Z dimension in depth with numbers. So remember, you're looking at collapsed Z dimension. Every 20 numbers is 20 sections, and that's 1 millimeter. So you get $1 after 20 numbers, 20 sections, 1 millimeter. So this is one animal, animal 7, and we're looking at 3 axons, 18, 19, and 20 from one injection site, injection in area V1 with a projection to area V2. And we could just concentrate here right now. If you look at the gross projection focus, it will look, yes, layer 4, very dense, that's fine. But if you were able to dissect individual arbors, and you can do that on the side of a focus, not through the dense portion, you're going to see variability. So one result is this would be a typical axon from V1, primary sensory cortex, and it's about 200 microns in diameter. This axon also has about 200 microns in diameter, but instead of one arbor, you have 2 and even 3 spatially dispersed arbors. In other words, this axon would be a delimited, maybe dedicated arbor, but this one is over a wider area. We don't know the exact postsynaptic pool for these different arbors. It's possible that one is a principal arbor and another is secondary. It need not be identical postsynaptic sets. This would be example of feed forward. Here is another of V2 to V4. It's a different area, but notice sort of the same size of arbors, but more typically here you're going to have multiple arbors, perhaps reflecting the broader topographic organization of those regions. So here is your summary. Feed forward in layer 4. There is okay, let me go. We're going to come back to something else in after this section. So feed forward, this would be your typical pattern, middle layer in multiple arbors. But if you go to single axon reconstruction, there is one region that we know so far where that is not completely true, and that is area V1. Here's your injection. Here is the trajectory of the axon, and here is the termination in extrastriate region MT or V5 at lower and higher magnification. This is very similar to what we had seen, layer 4, but they're 2 arbors, not identical. And look at this one with the arrow. Here you have not layer 4, but layer 6. So this bistratified termination pattern would never have been seen without the single axon. Even if you had Golgi-like delineation, you could not be sure that it was from one axon. This bistratified and multiple arbor termination may, one would like to think, correlates with the functional organization of MTV5. And in fact, there had been reports of a columnar organization in terms of directionality. Okay, now I said there are other systems. And this is the broad categorization of feedback connections. Not layer 4, but now a termination in layer 1. The cells of origin also are different. We're going to pass over that in this presentation. But layers, not layer 4, but layer 1. And the postsynaptic termination there, therefore, is probably the zone of distal apical dendrites, very different from relays through layer 4. The beginning of this was, again, with global injections. We could not see individual axons, so this is what was autoradiography, tritiated amino acids. And when we were working with this in the 80s, it was really quite glorious and exciting. Injection here was in MTV5, and you can see bands of label feedback to layer 1, in layer 1, 4B, and 6. This is white matter. This is another area to V2. But this could not tell you much about the spatial divergence of the spatial topography of those axons. When we moved to biocytin or BDA, all of a sudden you could see that. So this is typical termination of feedback ascending to the PO surface, usually in a straight line, which is a bit unusual, and then making an L-shaped turn. You can see differences in the caliber. These are, by the way, the neurons of origin in layer 6 and upper 3. And then with reconstruction, you can get a very, very clear idea of the divergence. So even these axons tend to be millimeter divergent in layer 1, sometimes with boutons due to synapses scattered over the length of the axon, as you could see here, but very frequently clustered in larger or smaller regions. But the take-home message here is of divergent in layer 1, and what we know less about the exact postsynaptic targets, but presumably concentrating on the zone of distal apical dendrites in that region. Not exclusively, about 20% probably are terminating interneurons. So there is a microcircuitry that still needs to be worked out. Let me comment on this axon. Here is the ascending portion of the main axon. The cell body is in extrastriate V4, termination in V1, ascending to the PO surface in layer 1, and then branching here, not as an L, but actually as a T, with what we sometimes call a hybrid organization. So here you have clusters, and then you have linear arrays of terminations. Same axon, but talking very differently to sets of neurons, either clustered or divergent in one area. Okay. I did not mention in the chapter, but I will briefly now, another system, which is the cortical pulvinar. Here you also have a duality from different layers, layer 5 in the cortex and layer 6. Layer 5 neurons send a small arbor, as you can see here, with large boutons that are clustered around the proximal portion of a neuron. Layer 6, which are more numerous, send small bouton extended arbors to the same region. So you can see here, this is from layer 5, low and higher magnification, and from layer 6, low and higher magnification, and these are the small bouton terminations. Now the small and large bouton dichotomy had been known for several years, first with electron microscopy, and then with other light microscopic techniques. But what single axon showed you was a striking difference in divergence. This is shown schematically here and in the photomicrograph. So always a delimited layer 5, small number, in conjunction from the same area with a spatially divergent termination from the other layer 6. And this is by analogy, and it's only analogy, reminiscent of feed forward delimited and feed back over a wide area. This is millimeters, this is microns. The return projection from the pulvinar to the cortex, we have fewer examples, but this also shows you a very complex arbor from terminating this time in layer 4, but also in layer 5 and 6. The remarkable thing here is the extent of this territory And it would be, your working hypothesis would be that you have delimited arbors that are efficacious here and then perhaps modulatory surround. In the article, I also mentioned in passing that single axons show you the trajectory. So sometimes we talk about shortest pathway in the cortex. You can easily find examples where axons do not take the shortest pathway. Here, they're backing onto themselves, terminating from V2 to V1, but then going back. And the same thing happens actually with geniculocortical parallel processes. And the last thing in terms of data, this I did not include in the report, but will mention. I said trajectory. You can also, of course, if you do reconstruction, look at fiber pathways. It's not necessarily the best technique because it's very labor intensive and slow, but it's something we should be able to address as the techniques improve with whole brain or slab terminations. So this is an example of what we might find. However, BDA labeled axons crossing in the corpus callosum from parietal injection. These are, of course, segments. And we reconstructed several sets of these axons, starting in the midline and then going back laterally. Let me see, we're not showing the injection site, but here is the corpus callosum going back laterally, 6 millimeters, 5 millimeters, and we color coded. So notice that the colors change. In other words, these are not it's not a box of spaghetti in the corpus callosum, but rather a bowl of twisted axons that are changing trajectory as they go through. So they start ordered, they end up ordered in the other hemisphere, but in the trajectory, they are shuffled. So in summary, I hope I have convinced you that single axons do give us data in terms of whether things are uniform or diverse. They're diverse and more than just accidental. It looks like there is some kind of organization. You can derive quantitative parameters. I didn't really give you examples, but we could go to the original papers. You can see collaterals. You can at least think about microcircuitry. And then in conclusion, let me mention Professor Zilles, who unfortunately, as we know, was deceased recently. He was a marvelous, marvelous person, a dear friend, and in the last years of his life, the last 5 years, he had worked on not single axons, but on polarized light imaging, PLI, where he and colleagues will continue this, were starting to dissect out fiber trajectories using the polarized light color polarimetry. And I think with that, I will stop.
Lovely. Thank you so much. Are there questions in the audience?
So can I ask Stefan?
Of course. Go ahead, Laszlo.
Kathy, you probably have the most experience. Can you just give some idea what about the columnar organization, which was also pinpointed in the 1SN original study, the feed-forward, feedback, and sort of lateral in primate versus in rodents, where you certainly can see, although the cortico-cortical connection are complicated, there could be trilaminar, but there are certainly columnar-like arborization. Now, when you see, of course, single axons in very detail, it looks like we are losing the big picture. Or can we go back to some, elementary big picture?
Okay, well, I think Laszlo, that's probably a different special topic, and I'm going to largely punt on that. But let me see. One comment certainly. Well, obviously, what I would say is a columnar organization probably is ripe for re-examination, and it's a very complicated issue. But in the context of what I was the small context that I was raising here. Not all connections are columnar, but if we accept for the time being the importance of columnarity and maybe even the existence of it, maybe, we still have to say, well, then there's cross-columnar. And that's not really too shocking because we've known for a long time that the neuromodulatory, dopaminergic, cholinergic, etc., will go cross-column. What is a little surprising to me, actually, is the divergence, the great distance that they go. And then coming back with some acknowledgement of columns, remember what I called hybrid axons, that there was an evidence even there in layer 1 for clustering and for divergent. So unfortunately, I can't give you more information on this or more data, but I think it would be interesting to think of this hybrid thing that even one axon can go in a columnar fashion and a non-columnar fashion. And I would say that same thing in the monkey when you have principal, and other people have mentioned this, principal and secondary arbors. I think another example that would be quite interesting to look at is collateralization. So what do the branched axons do in different areas? And because they're so huge, we have relatively little information on is it columnar in one area and divergent in another? Obviously, since I picked up the Klaska paper, I would favor the more interesting idea that you're going to have a divergent pattern from one axon in different areas, but I don't know. That the mouse, the only thing I would say here is, well, I'll punt again, that a lot of data will be coming out as people dig in to the whole brain images. Right now, we just have pictures, I think, very little data. Who was it? I'm sorry, one of the Japanese colleagues did go back in and reconstruct a high number of corticostriatal. But there, I'm going to sound maybe a little bit boring, but I'm going to say the same thing of diversity. And the result I would quote here is the Han, H-A-N, and Tony Zador with the broadcast and dedicated axons. So there are connections. They were not talking about column here, but simply the organization of the connections that V1 from one injection site would project via different axons. That was the point, that they could look at the different axons. And also with Tony Zador's map-seq, have a larger sample size. The downside of single axon is your miserably small sample size. And there they found axons that went to one extrastriate area, to 2, 3, I think even up to 7. So again, that's, I think you're looking at in part, 1, the actual data, and that's still in progress. 2, temperament, are you lumper or a splitter, for example? And I would be pretty much, I guess, a splitter in this category. Preferring to see diversity. So I'm sorry, that's probably a lot of words to say very little, but we can come back to this at some point.
Kathy.
Hi, Salim.
Hi, how are you? So we also showed columnar, the axonal projection from TUO to T, right?
Yeah.
Yeah. So it's still there, actually. So there's a columnar axons. We showed in temporal cortex, also from T to perirhinal. It's also columnar too. Some of the axons, not all of them.
Well, you see that, then I'm going to come back to the same thing of lumper or splitter. And yeah, we need to have a larger sample size and maybe multiple techniques as well. So I'm not going to say no, I'm just going to say that there's enough ambiguity that we could still take I don't want to say different size, but different perspectives.
Mhm. Yeah.
Are there any more questions?
Hi.
Yes.
So I think that many of the studies are focusing on maybe area-to-area connection, like maybe structure connection between B2 to B1 or parabiner to extra straight or whatever. But I think in the single axon approach, at least as you mentioned in the paper, maybe you can potentially see small intrinsic connection, like some connection within the same area. Then could you give us a little more about your perspective about what did you learn about some organization principle of intrinsic connection, or what these intrinsic connection will tell us about some functions?
Right. Okay. And as you kindly said, it would be from my perspective, and I would share with you mainly the papers over the years, but especially recently from the Kevin Martin group. And I think that's one of their pictures that I think I reproduce, or else I do in another recent paper that I had written for NeuroImage, I think. Okay, so I'm going to talk from Kevin's point of view and mainly say two things. One, he and Rodney Douglas, of course, called this the DAISY pinwheel. And the extraordinary thing is you really need intercellular fill for this. And we have a very small sample size, so we don't know what the rules are for how many collaterals you're going to have from different neurons. My guess is that it's obviously a range, and in my mind, it's sort of like maybe 9 to 10 from a pyramidal neuron in a plain vanilla cortical area. Now, Kevin also reported at the end, and that's why I became sensitized to it, a hybrid, what's really a hybrid arbor with, in fact, not only were there clustered and divergent pattern from one intrinsic collateral going over 2 millimeters or so, but some branches are myelinated, some are not, which I consider frankly shocking, that one local collateral, forget extrinsic, will have some of the collaterals myelinated and some not. Has to be saying something maybe in terms of timing or functional microcircuitry, subcellular specialization. Now, I know I've said very little on function because I think it's let me say it's still evolving, but Kevin's work and all of that and other colleagues using the same technique do give you a glimpse of how the intrinsic arbors, intrinsic branches might correlate with, of course, all of the, and here you do have the flagrant columnar organization of V1, so orientation columns, ocular dominance columns, and you can go on even with cytochrome oxidase. And there, I remember just barely touching on this in the 80s. Again, there were a little bit two schools of thought. The majority was that you had like to like. If a neuron was in a vertical column, it would terminate in vertical orientation columns. Jenny Lund and I always felt that was not quite true from our perspective, that you would get maybe 80% majority, but not always a significant difference of jitter. And then here I would go again to Kevin. I can't tell you exactly which paper, but there are only about 3 or 4 in the last 5 years, where he was also saying a little bit more complicated, not completely, not 100% like to like. And I think that's also the true for mouse, to go back to Laszlo's question there. The first results were emphasizing the like-to-like connectivity, but as you see work continuing with that, there are people talking about heterogeneity. So again, many words, maybe, certainly I didn't give you definitive, but that would be my perspective.
Thank you very much. Thank you.
Are there any other questions in the audience? Not that I can see. So I know that Michelle and I are dying to ask a couple of questions, but it's also time for the general discussion. So I would just hand over to Michelle, who's leading the discussion, and ask the questions. that are still missing. Over to you.
All right, so thank you very much. It was like a wonderful list of talks. And so I have like a couple of questions for Kathleen that I'm dying to ask. And I couldn't ask any question because I'm actually the mind behind like playing the recording, and I don't want to have any disturbance in the stream. Those are very simple questions, but you know, in the neuroimaging field, people are evolving in the bubble of neuroimaging, and we deriving sometimes for how true the neuroanatomy can be just because our mathematical model are showing us things that we want to believe. And it's so amazing to have the opportunity to speak with somebody like you who've been measuring and seeing really how neurons behave in the brain. So I have like some basic questions that I'd like you to answer. Like, for example, typically the axonal diameters that you can observe when you're doing those tracing, how large is the diameter of axons you're measuring?
Right, a beautiful question. If I were redoing or restarting this now, I would maybe be, hire people and work and concentrate on this. But the only large axons in cortical that I have passed through my hands are the V1 to MT. And that is probably, and there I don't know if it's from Meinert cells, would be my guess, or it's from could even be from 4B. With that exception, I would simplify a little bit, say they're all sort of about 1 millimeter. It probably is a range from 0.5 or 0.75 to 1.5, maybe 2. If you were looking at the cortical pulvinar, for example, you could see that the small Bhutan, which I'll call type 2, axons are a little bit larger and a little bit darker in the DAB than the type 1 with the small Bhutan. So you can see that difference. The actual, I measured under 100 micron, 100x objective, but I don't really, all I could say is be above 1 micron, below 1 micron probably, with the exception of V1 to MT, where not all of the axons. This is the other thing that is worth incorporating to my response. I think the trajectories are not completely uniform. You're going to have some thick, some thin, and some in the middle. And what's going to change if we had a larger database is the proportion.
Okay, because, like with diffusion, some team are pushing forward to the quantification of the average axon diameter in the white matter. Some others are saying it is not possible. And one of the talks that was presented today was showing in the core of the white matter an average of 7 microns. And I found it like a little high compared to what I read. And I wanted to have your feedback on that.
Yeah, it is. I'm sorry, that was in human though, wasn't it?
In humans, yeah.
Yeah, I still think 7 is a little bit high. Everything I gave you, of course, was from macaque, where 3 was conspicuously large. And the other thing, I can look, maybe not today, maybe not this week, but I wrote a chapter, which I can send you maybe, but sometimes like parvalbumin, we heard, maybe not, but parvalbumin will show the optic radiations. So you can go into the white matter with parvalbumin and calbindin stain, because in the primate, that will show you the thalamocortical, and you can get an idea of what the measurements are from that. In the monkey, it would also work in the human, but someone has to do a slab of sequential sections in the human. It's quite feasible, and I think it's really would be a nice exercise for someone that starts routine but would give data. There is a paper there's a book in the motor cortex. It must be Roger Lemon on talking about the caliber of cortical spinal, and I always forget this, but I think it's one of the better sources. It's a little bit old. And if I remember correctly, he says, well, they're not as large as you would think, but I can try to find that, or at least give you, and then send you those 2 things.
Oh, that'd be great. That'd be really great. Another question that I had is, so like the original depiction of the white matter from postmortem studies always stress how beautifully organized the white matter axon and pathways are in the brain. But you bring those single axon measurements that look like a little bit chaotic and doing like this straight turn and not always taking the most direct path to one point to another. And I was wondering whether, you know, those axons were isolated case or are like other axons hacking with those axons and following the same chaotic direction or trajectory in the brain. What would be your vision on that? Because that would change radically the way doing in vivo tractography.
And you have no idea, I mean, you probably do, how frustrated I am that, I mean, I left this work about 10 years ago, and you need sequential sections, you see. So the only thing you could address that with would be parvalbumin at this point, or maybe the folks working with expansion microscopy. What I can say is the examples I gave you were the subcortical white matter. So something that would be, I don't know really from those what happens in the track. That was all the distal portion, not the distal most, but you know, approaching and then turning. I don't think it's rare because, but it's not necessarily common. So you could say whatever you, unfortunately, you could say what you want. Sometimes I think I'd like to go back and look at least at the U fibers. This would be a project for a rainy day, which has not occurred yet. We had a whole series of beautiful days. But the other thing though, from my own data, the braids that I showed you in the corpus callosum, that I think is common. That's what they do. And it is a horrifying thought. I mean, they start topographic, they end topographic, and they don't seem to care in between. But there is a whole literature on, maybe it's from the development of the optic tract, where that same phenomenon is mentioned, and the mammillothalamic also. So I think it'd be a beautiful, probably a PhD thesis, enough for going into that, or validation. I've been talking with folks in Copenhagen, as you may know. There's plans to continue discussion on validation and dye attenuation. The PLI and dye attenuation may be a way to go for that as well. So excellent points. And all I can say is I wish I was starting over in 2000 or so. And we were doing other things, but maybe this would have been a precursor.
You know, I'd have a lot of other questions, but we'll probably follow up by email. I'd like to open a little bit more like the discussion to the floor. And with this, like there are like some two fundamental things that we can discuss. One, which is through studying connections, because this is Brain Structure and Function journal. What we're looking at is the integration of the activity of different regions. As we've seen in the harbor of those neurons, they collect information from different regions to project on other regions. And this looks like an integrative process in terms of function. And another thing that, you know, we can also discuss, and I've been thinking about watching all this lecture and being mute, was that some people use the connections to create gradient and erase completely boundaries between regions. Some others want to divide the brain in different regions and looking at the connection, how they project. To me, white matter connections and projection are not really a gradient. They have like finite borders, but you know, we can open this discussion and see what you guys and researchers think about this. I just killed the vibe.
No, I can say something. So yeah, so you're mentioning the whole cluster versus gradient, which I think which is a very fair discussion to have. But I think there's room for both, and I think you introduced it already. So I will agree with you that connection of a given white matter tract will probably cluster around a region. But I think there's still room for gradients for two reasons. So first, the global organization of the projection of these tracts, and not just looking at one specific tract, that's the one that we tend to find that is mostly gradual. And also the fact that if you look at some specific fibers, you can actually find gradients within them. So for example, if you look at like the optic radiation, for example, the projections of the optic radiation towards the primary visual cortex, for example, you could definitely find some structure there in which, let's say, now I don't recall exactly so there is this, if you make a cross section on the optic radiation, what we tend to find, for example, is that if you make that cross section, you will tend to see some sort of U shape in that cross section, in which the fibers that are most lateral on the optic radiation will project towards the back, so towards more posterior ends of the visual cortex, and the ones more medial will project towards most anterior portions of the visual cortex. So again, I think there is here. I think the whole cluster gradient discussion becomes a bit again becomes actually a bit, to pun not intended, a bit more gradual, right? At some point, you can say that yes, there is definitely you can definitely parcellate the brain in terms of which tracts hit a certain region, but first, the global organization of tract projection itself can be found to be gradual, and also gradual profiles can be seen within the white matter fibers themselves in particular examples, and I described one of them.
So, okay, so yes, in terms of, that's absolutely true. In terms of gradual, from my own experience, really projection tracks and maybe commercial tracks will show such gradients in the projection of the cortex, and will help to create those different gradient in the brain. But associative tracks, as far as I know, really, are really having kind of finite boundaries in the areas they project, and that will maybe reflect completely different mechanism of processing in the brain. Just so there I totally agree with you. It's again, I think it's more, if you look at one specific tracts and as specific territories that for a given tract, I think yes, you're definitely right. But then it's just more about if you look at the global collection of tracts, usually that's the one that we tend to see that is more gradual, and the shift between those is more gradual. Any other questions and remarks? See that everybody is a bit tired by these 2 hours of intense neuroanatomy and structural connections and methods. So in that case, I'd like to thank Brain Structure and Function for allowing us to do this wonderful special issue with Hiromasa Takemura, clinical neuroanatomy seminars, and especially Stephanie Forkel for helping us handling and organizing all these events, as well as the Springer Neuro Network. And I see Amy is connected here. She's taking care of all that. And I'd like to Invite you all to come back tomorrow from 4 to 6 to have the second session of a presentation about the paper of the special issue. We'll have a fantastic talk from Takaaki Kaneko on the spatial organization of the occipital white matter tract in the common marmoset. We'll have Michela Gamberini speaking about the structural connectivity and functional properties of the macaque superior parietal lobule. Dr. Quinn Andrews will we speak about brain structure and internalizing and externalizing behavior in typically developing children and adolescents. Stefano Ionanucci speaking about white matter correlates of hemiphase dominance in happy and sad expression. David Charles van der Zweven, sorry if I know, the role of diffusion tractography in refining glial tumor resections. And we'll finish up with a further discussion together tomorrow. Thank you very much, and I look forward to see you tomorrow for further talks.
Thank you.
Thank you.
Bye.
Yeah.
Bye.
Thank you.